In this part, we are talking about basic amino acids. Here, the way I learned about the basic amino acid is urgent lysis of basic history. So here, urgent means I'm talking about arginine. Lysis means lysine. Basic means basic amino acid. History means histamine. This is how histidine. So this is how I learned this. You can use different methods to learn this. Anyway, three amino acids are mainly basic amino acids. They are uh, arginine is there, lysine is there, histidine is there. So talking about arginine is a semi-essential amino acid. So what about lysine and histidine means? Lysine is an essential amino acid, but histidine is along with arginine, it's a semi-essential amino acid. So here lysine is essential, semi-essential, histidine is semi-essential. So anyway, talking about arginine, it's semi-essential, it's glucogenic alone. So that's all. That's all. But how arginine have another role there? In a way, we know that histones are something in which we know that DNA histones are basically 8 histone octamer is there, and DNA is rolled around it. This, we know the structure of we learn more in genetics, but we know how the DNA is rolled upon, because it's a long DNA. It's meters. We can't put directly into the it will be like what, messy like noodles, but we want to make into a basic structure. So histones are those which are those in which this DNA is rolled upon. So this is that the histones are made up basically with the structure of arginine. Arginine is a major component of histone, that's all. So histone rather than that, what are the other uses of histones? We are used for urea synthesis, used for nitric oxide synthesis, used for creatine synthesis. We all know creatine, we already learned just about that. Creatine means this GAM. GAM means glycine, arginine, methionine. Methionine section we will learn about creatine. Arginine section we should learn now, and glycine we already told about in the earlier section. So glycine, arginine, methionine, that's creatine. That's all. Creatine is gone. Urea is also used on the urea cycle. We are talking more about how urea, why urea, and all. Urea is basically, that's how from the urea, that's how we are. Why urea is formed is because that's how we are excreting this excess amount of nitrogenous compound in the body. We are converting it into urea. That process we need arginine. Arginine is one of the component of urea synthesis. So that's early urea cycle. That's another thing. So now here it's important is nitric oxide. Nitric oxide arginine, which is converted into citrulline with the help of a nitric oxide synthase. So thereby nitric oxide is released. So basically arginine is converted into citrulline. Citrulline is a basic part which is used for urea cycle. In that process, nitric oxide synthesis, this nitric oxide is a basic and very important vasodilator, vasodilator, and also that's along with vasodilation, it's also a relaxation of smooth muscles by releasing cyclic GMP, which is very important. So nitric oxide is a vasodilator along with a smooth muscle relaxation due to the process of formation of cyclic GMP. So anyway, it is that's a useful part of nitric oxide, which is formed from the arginine. Arginine is one of the that part. So moving on with lysine, that's the second basic amino acid. Talking about lysine, which is a precursor of carnitine. Carnitine is the one we are talking about, carnitine in electron transport chain. But here it's a precursor of carnitine. Lysine has a polyamine part. Polyamines of lysines are spermine and putrescine. That's another part. Lysine is essential amino acid. We have to take lysine, which is not produced in the body. So lysine won't undergo any transformation and all. Basically, it's used for carnitine. That's basic thing. So carnitine is a major part in ETC and all. So not yeah, and that's that. Moreover, that not ETC has such, but it's mainly for beta oxidation of fatty acid. That's a major part. And I'm talking about ETC. It's not basically ETC. It is just beta oxidation. That's what we are using. So last basic amino acid is histidine. Histidine is a basic amino acid which is semi-essential. Already told. It's a stable amino acid with an intracellular pH. At an intracellular pH, it's stable, and histidine is also a major part of heme. So talking about histidine, from the histidine we are producing histamine by just releasing carbon dioxide. We are taking out the carbon dioxide from the histidine to form histamine. It's a biogenic amine. That's all. Then talking the remaining part. So histidine, when uricase is an enzyme which is acting on urocanate will be formed. This urocanate with multiple steps will end up with FIGLU. FIGLU means 4 amino Formiminoglutamate acid, that is FIGLU. This is formed, FIGLU is formed. Why it is important? Because from the FIGLU we are end up with glutamate. So histidine to histamine, this carbon dioxide is released. Histidine to glutamate, multiple steps are found. From this another amino acid is glutamate, glutamic acid, glutamate is formed, which is also a non-essential amino acid. Why? Because from we can make glutamate from the histidine. No, histidine is semi-essential. It's partially formed and partially we can use it as an intake before. Why it is partially semi-essential? Because half of it can be used for the synthesis of glutamate also. So it is semi-essential in one way or another way. So glutamate become non-essential amino acid. So in this process of conversion of what histidine to glutamate, there is an intermediate called FIGLU. FIGLU from the FIGLU is converted to glutamate. So but in that step we need a B9, that's vitamin B9, and folic acid is important for that, because okay, that's a part. So from this what we know is that if vitamin B9 and folic acid is deficient, we will lack glutamic acid. Glutamate will be reduced. In another way, we can tell the histamine, the excess of the histamine will be converted into FIGLU, more FIGLU. There won't be glutamic acid, but FIGLU will be there. So if the patient is having excess FIGLU in the urine, that suggests that the patient is having a vitamin B9 deficiency, that is folic acid deficiency is there. The patient will having a excess FIGLU. So we all like there is a lot of correlation between different amino acids. I already told that same we are talking when we are talking about methionine, the amino acid methionine. I told that methionine is formed from the methylation of homocysteine. That methylation is given by is taken up by the methyl is taken up by the B9 or B, and that is transferred to B12. B12 is given to the homocysteine. So there, if B9 or B12 is deficient, the patient will have a homocystinuria. The patient will have excess of homocysteine and methionine will be less. So from there, homocysteine is increasing means and methionine is decreasing means that is what we are talking a non-classical form of homocystinuria. That's an important part where the deficiency is mainly B9 or B12. Here in B, if the FIGLU is more in the urine, that suggests that B9 is more. So different vitamins are acting as coenzyme in different parts of protein or amino acid metabolism. So different these here in these products, two things are important. What is histamine? What is FIGLU? These two things are important here in this process. Histamine, just it's decarboxylation process of histidine is called histamine. That's all. FIGLU is a byproduct, which is not a byproduct, that's an intermediate substrate in the formation of glutamic acid from the histamine, where this FIGLU is converted to glutamic acid in the final stage, where the vitamin B9 is essential. If vitamin B9 deficiency is there, FIGLU will be more in the urine. That's how we can differentiate vitamin B9 deficiency is there or not. So that's all about this clinical correlation is all, and this is what a clinical correlation is, I already told. So this is what we're talking about what basic amino acids.